Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. Quick note before we dive in. I was looking at our YouTube analytics the other day and saw that 93% of the people watching our videos aren't subscribed. If you're one of them and you value this work, take 5 seconds and hit subscribe. It helps us reach more scientists and lets us bring you more conversations like this one. Today, you'll meet Professor David Hodson, a veterinary surgeon turned metabolic physiologist who's redefining how we think about type 2 diabetes, obesity, and GPCRs. We'll talk about the long road from gut peptides to today's GLP-1 and GIP drugs, why receptor location in real tissue actually matters, and how fluorescent ligands are changing what we can see in the pancreas and brain. You'll also hear how collaboration with chemists like Dr. Johannes Brohagen and teams like Celtarys turn impossible receptor questions into real translational tools. What do drug discovery teams really mean when they talk about efficacy bias or functional selectivity? Terry's Corner unpacks the real-world meaning, one short, high-impact lesson at a time. Preview trailers are up now, so you can see exactly what you'll get. Links to Terry's Corner are in the description. Celtarys research is building the tools GPCR scientists need next. High-quality fluorescent ligands for dynamic real-time receptor assays. From kinetic binding to live cell readouts, their approach brings precision without radioactivity. We're proud to have them as a Dr. GPCR partner. Details about Celtarys research and their tools can also be found linked in the show notes. This episode is produced as part of our strategic partnership with Celtarys Research and is part one of a 3-part series of podcast episodes. Let's dive into this one now. Hello, everyone. This is Yamina from Dr. GPCR, and welcome to another edition of the Dr. GPCR podcast. This episode is special because we are recording an episode today with Professor David Hodson as part of a series of 3 episodes with our friends at Celtarys Research. And without further ado, David, welcome to the podcast.
Hi, how are you doing?
I am well. It's really nice to meet you, or actually see you for the second time. I'm really excited to be having this conversation today. So let's start at the beginning. And would you please introduce yourself? Tell us who you are and what is it that you do?
Yeah, so my name's David Hodson. I'm the Robert Turner Professor of Diabetic Medicine at the University of Oxford. And historically, my research has been type 2 diabetes. And more latterly, we've begun to research type 1 diabetes. And because of the type 2 diabetes connection, and because of the drugs that we'll no doubt talk about very soon, we do dabble a little bit in obesity just because of the sort of interconnectedness between the diseases.
Wonderful. I also have an icebreaker question before we dive into more of the research focus that you're working on. What's one thing about you that might surprise your students or colleagues?
I would like to think that I'm always a surprising individual, but I think somebody that surprises people is that actually I'm an inner geek and I really like aviation. And you don't want to travel to airports with me because I'm literally going to make you watch all the planes. I'm going to tell you all about the planes. And if you do travel with me, I'll probably try and take you on an interesting route on interesting aircraft types. So that's a surprising fact about me.
Thank you, David. Have you ever learned how to fly planes? Did you ever have an interest in doing that?
The thing is, I'm pretty highly myopic, so sort of my fear was my glasses come off in turbulence, I won't be able to find them, and then we're going to be in a bit of trouble, aren't we? So it's one of those things where I kind of like playing on flight sims and things like that, but I'm more passionate about kind of the facts and the figures and sort of, the engineering of it rather than the actual kind of flying. I'll let somebody else who's a bit better qualified do that.
Wonderful. Thank you for sharing that with us. Before we again dive into the research focus, I'd love it if you could walk us through your path to where you are today. I'd be curious to hear if David, the teenager, was ever fascinated by science in addition to planes.
Honestly, no, not really fascinated by science. I sort of grew up in the north of England. My family are pretty blue collar. I really wanted to kind of be an excavator, I think is the American term for it. We call it a digger here. I wanted to drive one of those because I'd sort of seen one out in the field and I thought it looked super cool. And then sort of what happened over time, because I sort of grew up in a rural area, I started to work a little bit on farms as a teenager, and then kind of saw veterinary surgeons kind of go about the business. Thought that looked pretty cool. And then, applied for and was admitted to train as a veterinary surgeon at veterinary school in the UK. So really, that's what got me into science, because of course you have to learn a lot of, basic physiology, pharmacology, you name it, you do all the basics in the first couple of years. And actually, weirdly, it was the pharmacology I liked the best, as well as the physiology, because to me that provided the explanation for how things worked. And we had some really passionate kind of lecturers at that point, who were actually, sort of taking all this time to teach a load of vets about sort of how acid-base balance in the blood and things like this. It was in the final year of veterinary school that I was faced with doing a surgery elective, which would have been a ton of on-call. But I noticed that the researchers in the building opposite were all kind of leaving at about 6 o'clock, whereas I was working through all night, basically. So I thought, I'll do that, and then I can concentrate on my exams, and I can sort of get a high grade, which will be helpful later on. And I actually did an immunology elective. We were studying mucosal immunology in Minnesota mini pigs. And then from that, I started to do a bit of work on anesthesia in mini pigs during some of the initial surgical approaches for laryngeal transplantation. And it really captivated me, because at that point I was working together with a human surgeon, an ear, nose and throat surgeon. And, just like seeing that individual there, taking time out of his evenings, weekends to sort of do these studies was, pretty inspiring, actually. And as kind of the bug bit, I was offered PhDs in immunology or in reproductive physiology. I hadn't massively listened to immunology as a vet student, because it just was super complicated. Sort of wish I had a little bit more now, but I sort of figured I could just kind of wing it on that one. But I was quite interested in reproductive physiology, simply because if I went back and worked as a veterinary surgeon, it was going to enable me to become a specialist in sort of farm animal kind of reproductive medicine, which is really key to production animal work. So I had a fantastic mentor during my PhD, Domingo Torcanesi, really taught me how to write, do stats, and all the rest of it. And then he sort of encouraged me to go to a postdoc. I went to a postdoc in Neuroendocrinology, which is the fancy word for the kind of bit of the brain which interfaces between the brain and the periphery. And unbeknownst to me, there was a lot of work going on in that kind of neuroendocrinology field in terms of food intake, appetite regulation, and so forth. But I was working on a parallel track. We were actually working on growth hormone and how it's regulated. And then at the end of that, with the methodology we learned, which was a lot of kind of advanced imaging at that time, I thought, okay, I want to get more into a translational kind of disease now, because growth hormone, deficits and overproduction are serious. Don't get me wrong, but I felt as though it was a little bit more limited compared to working in diabetes. So I went to work with Guy Rutter, who was an eminent islet biologist at that time, and he sort of trained me up in islet biology, and that kind of is where all the work on GPCRs came from, because it turns out that the brain is a great place for GPCRs, but it's not super accessible. Hex cells are a great place to look at GPCRs, but it's a hex cell. Whereas the islet kind of is a halfway house that you can isolate enough of them that you can really study GPCR signaling in detail, but you don't have the limitations of the brain, which is super challenging to culture and all the rest of it. So it turned out to be a really good model for looking at GPCR signaling in complex tissue, and that sort of stayed with us, and we still look at GPCRs now, but I wouldn't call myself a GPCR biologist. I'm not like hardcore enough to sort of do that, but we do work at the interface between sort of type 2 diabetes, obesity, and pharmacology as well. But as you will know, as long as you work with the right people, you can generally answer the right questions.
Thank you, David, for that. I think two things came into mind while you were walking us through your training and how you got to where you are today. I think you're the first ever guest who wanted to go to veterinary school. We've had people who wanted to go to med school. We've had people who wanted to be, astrophysicists, but never anyone has wanted went to veterinary school. And the second thing is that just the way you were explaining your trajectory, it became clear to me your interest in figuring out how things work, and that connects very well to biology as much as it connects to how planes work and the intricacies of their engineering.
Yeah, I mean, what I will say is that my parents remain highly confused as to why I'm doing any of this after having trained as a veterinary surgeon. And I think my bank account is probably less well off for it. But I think it's a good example that I think we're actually quite young when we have to make these decisions. And, people say, why did you not want to be a clinician? Well, I just don't like dealing with humans. Okay, like I'm fine with it, but human medicine never was a thing for me. So I think that's a great thing about if you do anything that's allied to science, is that there's always a lot of possibilities that come from that, right? So yeah, point taken.
No, I agree. I absolutely agree. And I think to your point, I think we're made to be taking these decisions very early on. And sometimes you need time to mature, but sometimes you also need time to try things out. So having the opportunity to, what I'm thinking about training of, scientists in the lab, when you have the opportunity to work in different labs, it allows you to get a taste of what kind of research can be done and find your path. And I think this is pretty much what happened to you, is that you ended up working in the lab. You picked the lab for a specific reason. But then you found a passion, and here we are today. Wonderful. You walked us through your path, and I guess that's what a little bit motivated you or got you towards working on type 2 diabetes and obesity. But can you maybe tell us a little bit more about the moment where you realized, oh, actually, I'm very interested in this field, and this is something I really want to pursue?
So the thing with type 2 diabetes is that you will know somebody that has type 2 diabetes, right? So it's something that will, have impact across society. It's obviously a bit of what we call a silent disease, because you might not necessarily know that unless somebody explicitly says that. But the reality is that if you have type 2 diabetes, on average, and these are reasonably old data now, you would live 10 years less long than somebody without type 2 diabetes. And there's quite a sharp sort of socioeconomic split, right? And I think that's particularly acute in the US, right? So it's those that can afford the medication, those that can't afford the medication. And although medication is all free in the UK, you just pay a prescription charge, we still have that disparity. So, and I'm not saying that I'm sort of, a fighter for justice and equality, but I don't like inequalities. And I think, this disease seemed to me to be quite important to investigate, just due to the impact that it has. I think, do you ever make the right decision, right? I mean, it's kind of like, I think as a researcher, one has to remain curious about things and follow data where they go, even if that takes you outside of the system that you're comfortable with. But ultimately, you sort of need to just have that kind of firm anchor that you can come back to. So in a way, kind of diabetes is my safety net when I get a bit scared of, GPCR and sort of, kind of brain work. So, it's a nice area to work in, because you can actually have positive impact with the work that you do. And, of course, it's very topical at the moment, because we went through many years without having massively effective therapies to suddenly having a therapy which is very effective. But even then, if you take that therapy, at some point it'll stop working, and and things will progress. So we should never kind of rest on our laurels, and we should always be looking to have the best possible treatment options. And that comes from just doing good, rigorous research.
No, I agree. I absolutely agree. My next question is I mean, I think everybody by now, especially nowadays, kind of knows the answer to that, but I really want to hear your perspective, and maybe a time, how things have evolved to discuss how GPCRs fit into the bigger picture of metabolic health. When I was at a meeting last week where there was a panel discussion, and it was about the GLP drugs and the incretin system, and treating or having peptides being used as drugs. And I remember someone on the panel mentioning, well, a couple of, 10 years ago, 15 years ago, people said, well, we will never have peptides as drugs. And look at where we are today. So from your perspective, in the context of metabolic health, when do GPCRs enter the scene, and how are they, ranked among other targets today?
So let's go back all the way to actually understanding how GPCRs might make a contribution. So using diabetes as an example, we have these massive prospective cohorts, and in the UK it was very well recorded because we have a national health system. So all the HbA1cs, or back at the time, blood glucose levels were monitored. Yet there was still no consensus even back in the sort of 80s about how you treat a patient. So my predecessor, Robert Turner, I'm the Robert Turner Professor now, that's sort of how important that individual was, did all the initial studies called the UK Prospective Diabetes Study, showing that the more aggressive you lower glucose levels, the better that patient does, which seems completely intuitive, but wasn't the prevailing thought at that time. And that showed that sulfonylureas, metformin, and insulin, are all very good to use. Now, the problem with all those drugs is that, particularly with metformin and sulfonylurea, is you sort of have this 7-year timeline where they work and then they stop working and then you need to escalate treatments. So in parallel to all this, and I'm standing on the shoulders of giants here, there was a load of work going on in gut peptides. And gut peptides at the time were these kind of things that were released and influenced, kind of appetite and things like that. And the system wasn't very tractable. And the incretin effect was sort of well documented by Creutzfeldt very early on as being, when you eat food, you get this large spike in gut hormone secretion, specifically GLP-1 and GIP, and that then boosts insulin. And actually, it accounts for about 70% of your insulin production. So we always knew that these were really, kind of potent hormones. But like you say, it was quite difficult linking a peptide. And that's the key, right? But before we knew that peptides could be therapeutic candidates, well, most ligands for GPCRs are peptides. So that then naturally brought peptides in as being a target. And I think GPCRs, people sort of, beta-adrenergic and all sort of the early kind of hits became incredibly important. But as a drug for metabolism, we were sort of lagging behind it, even though we knew all the underpinnings and that these things had targets that were GPCRs. So it was about 40 years of work, probably more, about 50 actually, starting with GIP and then GLP-1, all the way through from cloning the genes to then having to get a stabilized analog. And it's a great, read the stories about it because it goes all the way from sort of, anglerfish to lizards, through to human trials. And it's a really nice story about how multiple pieces and strands of evidence need to be built to make, really impactful drugs. And, the issue with peptides is degradation, right?
Yeah.
So in our particular case, GLP-1 is degraded within minutes by dipeptidizes in the blood. So it was just no good as a therapy. Like my stomach's producing it all the time and I eat, that's fine, right? I'm my own endogenous producer of that. But your capacity gets overwhelmed during type 2 diabetes to do that. So the key was really figuring out how to modify those peptides so that they were stable as agonists. And I think that then sort of exploded sort of all the GPCRs as metabolic targets fields. Sort of remiss as well because I've got tons of colleagues working on, human genetics of this as well, but just with a focus on class B GPCRs, particularly the secretin receptor family, so and also the glucagon receptor family. This has now brought this into the absolute, mainstay of first type 2 diabetes treatment, now obesity, and then kind of going forwards into other diseases. So I think it's a really interesting time to be a GPCR signaling person, because I think that we are now beginning to see all the pharmacology that was done and all the amazing work that was being done really pulling through into just really important treatments.
Thank you for that. I think it makes it's a fascinating step forward. And while you were mentioning this, I was thinking, it took 50 years to get here, but with the avenue of AI and of tools and collaboration tools, I wonder how long it'll take to gather the next set of data in the context of type 2 diabetes or obesity or any other disease in order to make that leap. So if we can hopefully use all these tools and use AI and then reduce that discovery time or the next milestone by half, it will be already fantastic.
Agreed. I think you sort of have to, back then when they cloned the pro-glucagon gene, which then was how they gave rise to understanding glucagon-like peptide 1. I wrote an article recently called Classics in Diabetes, and I wrote that article together with the person that first cloned the GLP-1 receptor gene. Well, we can now do that like in a day. We can even just pay someone to do it quickly. Yeah. That was, years of work at that point. And before that, they didn't have any way to recombinantly overexpress, to sort of do these studies. So what I mean by that, it doesn't matter what tools we have. If you look at the work that came before the GLP-1 receptor was cloned, they were still figuring out that, N-terminal truncation was leading to antagonist activity. Yeah. It's just everything was a bit slower through. But so I think it's helpful to have those things. And I think sort of as we now have therapies for most diseases and we look to have more refined or personalized therapies, that will come more to the fore. But you can't get around the fact that if you want to get a drug to market to treat a population as large as the population that has diabetes or obesity, FDA and other regulatory bodies have very set standards on how you go about that. And it's billions, if not hundreds of billions of dollars of investment to get to that point. So to make a new drug from scratch, like I say, it might be helpful to have those things to help you get the target more refined, to sort of get rid of off targets, but the process still remains the same underneath. So I think it's going to be interesting to see how they all come in. I mean, obviously the game changer for us has been Cryo-EM, because before Cryo-EM it was hard to figure out exactly what was happening. But we're now seeing Cryo-EM lessons sort of pull forwards as well. So I just think it takes a range of things, but ultimately the buck stops with the big pharma who are going to pay the money to do the testing, right?
Wonderful. No, absolutely, I agree. And to add on to the Cryo-EM comment you had, I think MD simulations are going to be are already taking that space. It's great to have a Cryo-EM structure, but it's a snapshot in time. So being able to simulate the different states, the different conformational changes in the presence and absence of peptides or any ligands will become very important to get a better sense of what is going on with your receptor in the presence of that.
It's a mega technique, right? I mean, I've just spent 8 years working on something, and then we managed to get a low resolution cryo structure. We're trying to get the higher cryo structure, and it explains everything. It's like, well, if I'd just done this before, then I might have saved 8 years, but actually would have never got to that point. So I think it's a really good way of just confirming, how valuable your data might be going forwards. But yeah, it's MD simulations. It's similarly important.
Absolutely. Speaking of tools, we're talking today because Maria Mayalaro from Celtarys Research and Wilson Gomez suggested we record this 3-episode series about your research, your collaborators, chemical tools, and Celtarys Research. So I want to deep dive into where did that partnership start from? And I know you collaborate with JB's team, and you use their know-how in chemistry in order to be able to visualize what happens with the receptor in that context. So tell us a little bit more about the importance of collaboration in science, and specifically with this collaboration.
Yeah, so I mean, I don't know about other people listening to the podcast, but I'm simply just don't have the ability to achieve the research we do with a whole ton of collaborators helping out. And I think we're at a stage now in science where things are quite specialist. So, you want to do some human genetics. Okay, let's go to the human genetics people. Want to do some food intake studies. Let's go to the people that have the cages, the ethics, and the knowledge. And that, it's really cool to be able to do that. And people are really, open to do that. A sort of back in the day when you had too many authors on your paper, people would be like, well, what is it you're doing yourself? Whereas now, when I don't see enough authors, I'm thinking, how have you done that? And, who's reconfirmed, the data that's come through. So I always think about it as being like a sense checking thing as well, because I can't ask collaborators to do things without them reconfirming it and then, sort of going ahead and investing the money. But also it just gives you this massive capability that I don't think any single individual is capable of. I mean, there's only one of me, right? So it's a time limitation. I simply just can't sit here. And, as much as I love science, right, I've got other hobbies and I want to be out the door by 6 o'clock and not really worrying too much about sort of, killing myself to midnight every day. So it helps to balance as well. So that's why collaboration is important, right? And it's particularly important for interdisciplinary. Your other question was about how did I kind of collaborate with JB, right?
Yeah.
So when I was an assistant professor, I was living in London, and I got an email off a chemist called Dirk Treiner, who's now at UPenn. And Dirk Treiner's a big synthetic chemist. He's done all the major hits like sort of synthesizing tetrodotoxin and really kind of elegant synthetic challenges like that I know absolutely nothing about. And I'd sort of got chatting to him at a conference, and he was working on ATP sensitive potassium channels, so that was ion channels. And they had a tool which would light activate the channel. And this was at the time everybody was getting into photopharmacology, it was called. So he said, I'll tell you what, I'll send a student over to your lab for about 6 months, as it turned out. So, it was JB that turned up at my door. Ironically, as I was sort of doing some DIY in the house, fitting a new toilet, as it were. So I was in a pair of rubber gloves, pair of shorts, and a vest when he turned up, because obviously I was like, I don't want to get covered in sort of toilet backlash. So it was a strange introduction, but over time we sort of dug out a few papers on photopharmacology. Really kind of enjoyed working with one another as JB's career kind of developed, and obviously, I was really kind of pleased about that, and my career was developing at the same time. And we just kind of become good friends as well. And there's no clashes because I've got sort of my own kind of diabetes stuff. I'm not super territorial anyway. I think kind of more the merrier is better. But we just do things we wouldn't have individually done without the collaboration. But also the way I think and the way JB thinks is slightly opposite. So it's just helpful to kind of get those different viewpoints. And you'll see, a lot of people that listen to the podcast will have really close collaborators, right? And it's generally just people they want to work with and people that they enjoy working with. We're not here on Earth long enough to sort of, suffer for 30 years working with people that we don't. And then we'll probably drop the collaborators that are a little bit more stressful. But that team science approach, I think, is really critical. So, yeah, that's sort of how I met JB. And then things have evolved from, working on ion channels to sort of hitting GPCRs. And who knows what will come in the future. Maybe something else. So, it is not like it happened on purpose. It was also serendipitous and we built up around it. But I'm sure other people directly approach people and get great collaborations as well.
And you make a good point about making sure that whoever you collaborate with can contribute, but also it's a pleasant experience for everyone.
Yeah, I don't know. I'm not particularly old. Maybe I'm just not very patient, but we're very collaborative as a lab. And I just feel as though it allows me to do things that are of interest. So it just keeps me going. And I think it just comes down to the fact that as a researcher, right, you've got a choice to do other things and you can definitely earn more money and you can do all the stuff like that, but it's got to be a passion. And I think when it's your passion, like I sort of thought, why am I writing this paper on hardcore metabolism in beta cells, which I absolutely hated? Turned out to be a really good paper. And, now I look back at it, I'm quite proud of it. But at the time I didn't like it. And then I sort of told myself, well, isn't that fun that you're able to kind of learn new things every day and sort of, kind of push those boundaries a little bit and push yourself and prove to yourself that you can do it. And I think collaborators actually kind of make you do that as well, right? Because you sometimes got to get your head around a bit of chemistry or around a bit of, structural work. And I think, it sort of gets you out of your comfort zone. And I think that you should always be slightly working out of your comfort zone, to be honest, as a researcher.
As a human as well, I think, not only as a researcher, but yeah, I think growth happens when you're outside of your comfort zone and being able to venture out of that. That's how you grow and how you learn new things, whether you're a scientist or otherwise. All right. So can you share, speaking of your collaboration with JB and his team, can you share a moment when JB's tools gave an answer that you couldn't have gotten otherwise? Any highlights?
Well, I mean, yeah, I think the big thing for us is we do a lot of imaging, right? So we can use agonists, antagonists, we can look at cyclic AMP responses, but within a complex tissue, for example, in the islet, you can, sort of put an antagonist of the GLP-1 receptor on there and look what happens. But the ligand can be coming from an alpha cell, there can be an indirect interaction with a delta cell. So you can't really precise it down to being, okay, this is directly binding the receptor on the beta cell. You can take a knockout and make a conditional knockout, so you knock it out in the beta cell, and that's going to sort of get a bit better. But I've always felt as though you kind of got to see your target, and if you can't see your target, then I don't know, it just seems a bit, like it could be there, but it's just nice to sort of see it up front. Also, the question of how drugs actually access the target, right? We chuck something onto a pharmacological assay. We're doing like a cyclic AMP EC50. It's fine, the cells are all going to see it. But the moment you put a drug into the body, we don't know where it goes, what it binds to. And I think it's quite important to know. Some people, for example, with GLP-1 receptor agonists think that the more it goes into the brain, the more effective it's going to be. But that's not actually based upon any understanding of where the GLP-1 receptor and GIP receptor might be in the brain. And it turns out that actually brain penetration doesn't seem to be the biggest differentiator of therapeutic efficacy. It seems to actually be, which neurons are targeted, whether it's neurons that have both receptors, neurons that have other each receptor, or even supporting cell types like tiny sites. So I think we just wanted to be able to kind of see that in action, and this is what these tools have allowed us to do. And we've done a lot of testing on the tools. We've validated them using all the gold standard approaches. We've made knockouts, etc., etc. And then it really just comes down to many years of us working with antibodies. And then all GPCR people will know that antibodies against GPCR should immediately be treated with suspicion. And it was a lot of work by Dan Drucker's team in Toronto and Lottie Knudsen at Novo Nordisk that really showed how many studies there were using non-specific antibodies, but also produced some of the early antibodies that worked. But even then, the antibodies now are a little bit more effective, but they're not going to tell you where the drug's accessed. They're not going to sort of allow you to look in live tissue and answer all these questions. So I think really a lot of the tools we made were born out of kind of frustrations with just the conventional approaches, allowing us to look at these things. We're sort of taking that to the next level now with sort of snap tag knock-ins and so forth, and that's useful for now looking at different receptor pools, looking at receptor trafficking in complex tissue and so forth. So I guess in summary, what it's allowed us to do is visualize where the receptors are, both in the periphery and brain, understand the cellular substrates for some of these drugs. So where is it they're binding, which then allows us to explain some of their efficacy. And I think just ultimately have a tool that's well validated, that actually we've sent to kind of hundreds of labs now who are all using these tools to dig out insight into GLP-1 receptor signaling. So, labs working on platelets and bleeding disorders, other ones working on hypertension. And we know that the tools are specific, so we know that the data they will publish will have, translational impact with none of the waste. And I think actually that's one of the satisfying things. It's quite amusing because maybe JB will tell you all about this in his sort of next podcast, but these things were not designed on purpose. We got, did you use the term cocky in the US? Like we got a bit ahead of ourselves, right? So we thought, okay, let's make a photo switchable thing to illuminate the receptor to then, allow us to kind of optically control the receptor. And it was just really hard to do. And we had another student, Tom, who was working with us on this. So these just came as sort of what we would say is kind of like a project that didn't seem like it was leading to anything, and we just had to get something out. And it turns out to have actually been really important, for both of our work going forwards, actually. So, sometimes the things you think are going to end up on the cutting room floor can it's not the best work, right?
That's wonderful. And to your point, I think sometimes you have to follow your gut. You have to try out things, and if it doesn't work, now you know that it doesn't work, and then you can learn from it and then apply the learnings towards another project.
Or as they say, there's no shame in failure, right? Which is going to be quite a lot of your career in research. But yeah, I mean, I think that's something I think the funders need to be a little bit better about. Is we all have tons of negative data or stories that never really got off the ground because we were just, maybe the antibody's not specific. We have doubts, and I'm sort of in a position now where we have multiple things, so we can buffer it. But if I think back to being an assistant professor, it would have been like ruinous, right? And I think, just more support from funders that actually producing negative data or just debunking problematic reagents or problematic papers is totally worthwhile expenditure of grant income, right? Because, you can't estimate how damaging it is when somebody's not actually, doing something that's, going to make a difference later on.
Exactly. And I agree with you. And I think from a funder's perspective, but also from a publication perspective, when I was, I think, a PhD student, at some point I hit the streak of 6 months of no data or nothing positive. And I was joking around with one of my colleagues saying that we should establish the Journal of Negative Results and start gathering that information, because it saves everyone time and money to know what doesn't work.
100% agree. Yeah. And they have more impact.
I think so too. I think knowing what doesn't work is as important as knowing what works. It's two sides of the same coin in that sense.
It's kind of weird, isn't it? I mean, you could buy an antibody and you look at the validation, you're like, oh, no, this is not going to be good. But you buy it anyway. But imagine I bought a new car and it turned out actually it's not really a car. It's just like a ride-on lawnmower. I don't know how they get away with it, if I'm going to be honest with you, that all the workload falls onto us. And that's why I think the kind of initiatives like these kind of antibody repositories where they're looking at the rigor, they're looking at KO validation and all these sort of gold standard approaches are really valuable, because it just stops people wasting their time with, I think, companies that frankly have to and need to do better. Like, I'm not saying you get an antibody, GLP-1 receptor antibody doesn't work very well in the brain. There's a lot of tissue specificity. Yeah. It just doesn't work, but it is specific, right? Yeah. So a bit of a balance, but yeah, I think the GPCR field is pretty good for that actually, because I think because of the nature of the work, people tend to share the tools around and out, and everybody sort of knows, what works and what doesn't work.
Exactly. And I don't know if you're familiar with it, the Human Protein Atlas, a company called Luminex, and my former postdoc supervisor from Rockefeller University, Tom Sackmar, spent a lot of time looking at all of these antibodies and in a multiplex fashion to see how specific these antibodies were or not. I think they also published a paper on it. And there was a resource that I can find and share with you where they were focused at exactly this, figuring out of the ton of anti-GPCR antibodies out there on the market, which ones actually work and can detect specifically the target or not. And it's difficult as well to raise antibodies against GPCRs, but to your point, there needs to be a validation step.
You've got to validate them, and they'll have different sensitivities and specificities. Some will work for facts, some will work for immuno. If you see a histopathologist, they've validated all their antibodies on the histopathological panel for disease X or Y. And back in the day, that would have been pre-absorption, controls and all the rest of it. Now we have the luxury of knockouts, but I think for your purpose, you still have to validate it. But I agree that the human protein, human tissue atlas are really going to great pains to look at good antibodies. In fact, we've had the lowest failure rate with the atlas antibodies, which are the tandem mass spec and GTEC sort of kind of validated antibodies of all antibodies that we've used. We know who the usual sort of suspects are, but even that, they have some winners as well, so it's fine.
No, I agree. And I've been in situations where we needed to find an antibody against a target, and we went out and ordered everything that was available for the application we wanted to, and then just empirically spend the time to test it and see which one worked, and ended up with 0 at the end of the day. But we had to try it. All right. Any memorable aha moments in your lab where the data finally made sense? Anything that shaped the way you think about type 2 diabetes and metabolism in the context of GPCRs?
Yeah, we actually had one recently. I mean, I'm not really an aha person, right? I'm sort of like a born skeptic about most things. So I'm just sort of like, or maybe I'm just a bit slow and it takes me a little bit of time to sort of catch up and figure out what the importance might be. Yeah, I'm also aware of our limitations, to be fair, as well. But we've been working on something, a product derived from an alpha cell, which is in the pancreas, and the alpha cells release glucagon. And we've sort of been working on this kind of gene, kind of signature marker and signature protein for sort of 10 years now. And it's called vitamin D binding protein, a GC globulin, and we've worked on it in terms of how it regulates glucagon secretion. And we had a big aha moment, sort of last week, really. Last month maybe now, because we realized that actually this stuff is interacting with G protein coupled receptors, and explained a lot of the data that we were getting in terms of how alpha cells regulate beta cells by engaging GLP-1 receptor and so forth. And it was not until all this data was like, why is it stopped working on the GLP-1 receptor? How can we pull it down on co-IPs with the GLP-1 receptor? And it was just not making any sense because it's not an orthosteric ligand. And obviously we got the Cryo-EM structure with JB and JB's colleagues, Daniel. And it was like, right, that's what's happening. And it explained everything. And that was really satisfying, actually, because I almost shelved that project multiple times because I just didn't trust the data. I was like, what is going on? But we had multiple streams of data. And, we were like, is this fatty acids? What's happening here? So it was an aha moment. And, I doubt I'll get another one like that in my career. But on the flip side, I just feel very grateful to sort of be able to come in and just, kind of look through data, sort of go downstairs, see the imaging. So it sounds like corny or cheesy, I guess, as you might say in sort of the US, but I really, really enjoy the research, right? I think the stuff that bugs me more about academia is like the kind of admin and, the other things that you perceive get in the way of doing the research. But for research for me is like, a total luxury. So whatever the paper is, whatever the impact of the journal is, I think we always get really excited about it.
I think it's exciting as well when you get to sit down and just, read a paper or look at the data. And I think the most exciting part is getting in front of a plate reader and looking at the numbers and having your plate map and saying, okay, this molecule does this, that molecule does that compared to the control. And just being able to appreciate, for me at least, the shifts and curves is just, it gives me goosebumps and tears in my eyes every time I see something that is so beautiful. When you think about it, you're translating something happening in a cell and assigning numbers to it and being able to manipulate it. I think it's just fantastic. So David, how do you see the biggest opportunities for GPCR-targeted therapies in metabolic diseases? It's a big thing. Everybody's working on this, big pharma, small biotech. But in your opinion, what are we looking at as an opportunity perspective?
Tricky one, right? Because we've just been having a conversation about how, drugging GPCRs has been difficult because, peptides were never really seen as a conventional therapeutic, and now we have oral formulations, and people seem to be happy injecting, for specific diseases. And now, with the new GLP-1 receptor, GIP receptor drugs, such as Maritide, for example, it's a once monthly dosing. So, I think there's a lot of formulation challenges that go with that as well. But ultimately, we're in a good place, right? The drugs are effective, they work, right? And they're now beginning to sort of have impact in other disease states. So in the UK, for example, you're now able to give a GLP-1 receptor agonist, even if the individual has no diabetes or no obesity, but if they have, cardiovascular disease, because they can be protected for that. So, it's a really exciting time, and I think we kind of, have to follow patients up for longer to understand the true impact, but for sure, it's going to be very positive. I think if we just look at the number of people that have metabolic disease like type 2 and obesity, it's, well in the billions, right? And for all the people that can be treated and do well on the drugs, and there's going to be a lot that don't tolerate the drugs well. So I think the first thing is to kind of dial in the side effect profiles and figure out why some people suffer more side effects than others. I think the second problem is that if you stop certainly on the obesity drugs, there will be a rebound. Depends on the drug. Again, Maritide has less of a rebound. I think that in itself is worth studying, because why does this drug, which is only different because it has a GIP receptor antagonist, sort of have less of a rebound than a drug that has a GLP-1 receptor, GIP receptor agonist component? Because I think that's some really interesting biology. I think that we need to kind of, what a healthcare provider is going to do, right? Are we going to keep people on these drugs lifelong? They're going to come off patent, they're going to get cheaper. If we keep them on the drugs lifelong, what will the side effects be then? So we need very rigorous follow-up. And I think the big sort of thing that was a problem recently is maybe people are losing lean muscle mass. Will that predispose them to kind of, frailty and other issues going forward? So, do we now begin to look at muscle mass sparing compounds, and how does that look from a GPCR perspective? I mean, the activin combination recently has been paused, and that was one way of, having an activin receptor, and then the obviously GLP-1 receptor engaged to preserve muscle mass. Is it going to be myostatin going forward? Does it really matter, right? If you're not got metabolic disease and you lose some muscle mass and you're more active, is it okay? So I think there's lots of things to figure out, and there's this whole space now kind of in the conjugate field where, GPCRs are really useful as quite cell-specific and tissue-specific addresses. So what I mean by that, if you look at GLP-1 receptors, very specific for beta cells in the pancreas, so specific that you can use the GLP-1 receptor to sort cells in the pancreas, and it's basically 98% insulin positivity, so beta cells. So we use that as a tool frequently for that means. But you can also do the opposite, where you can make a GLP-1 receptor agonist. You can put a gene therapy on there and get it in the cell, and that's now cell-specific, right? So I think that's very exciting technology going forward. And then you look at other colleagues in Copenhagen that are looking on parallel pathways. So now we're taking a GPCR, we're taking some human genetics which support, for example, NRK2 or whatever it's called, involvement in, food intake and weight loss. And now we can combine the efficacy of both molecules in a safe manner. So it comes about sort of using all the molecules which were on the kind of cusp of that sort of therapeutic window and index, but now actually we can get the full therapeutic efficacy out of them by combining them. So, I had a grant that kind of sank because he said, well, the field's already mature, right? You got GLP-1, so why do we need to know more about that? Well, I mean, I think we don't look at cancer like that, do we? We don't say, okay, we don't want to make better chemotherapeutics that lead to better, patient outcomes. I think we should always be focused on the patient, and I think that going forward, we're now at the first phase, right? And we've achieved the weight loss that's needed, and that's remarkable. But there's a lot of different stuff to dial in. and a lot of patients that would benefit from that, and ultimately the patient outcome will be better. And ultimately what we want to do is, make patients not have the disease lifelong. But that's probably going to be difficult because, of course, as you age and you get multiple morbidities, things become a bit more difficult. So, I think it's tremendously exciting. I think obviously there's a very kind of metabolic GPCR slant on that, but I think this goes across GPCRs. I think it's been a profoundly impactful field, right? And it's really beginning to pull through now, which is probably nice for a lot of my colleagues to see. I've only been working in it 10 years, so I can't claim to having done any of the seminal pivotal work.
You were mentioning at the beginning, thank you for that. You were mentioning at the beginning that you don't consider yourself as a GPCR, hardcore GPCR. I think you're a GPCR physiologist. That's the term that came into mind. And to your point, being able, with what we know, we need more data as to how these drugs affect patients. And I'd add to that, one thing that usually, doctors and usually people don't measure, for example, the level of activity a patient has, the type of exercises that they do, the food intake, what kind of foods do they have, what is their genetic background. I'd be curious to see why some of these therapies work on certain population of patients, but none on others. And I think it's a complex thing to be measuring. And I hope one day we'll be able to put all of this information together in order to have a very targeted profile for these drugs and say, well, this is your genetic, this is how many calories your body burns, this is the type of exercise you do, this is the kind of food you intake, therefore this is the drug that you should be taking in order to help you for a limited amount of time to help your body rebound.
Yeah, I mean, there's some really good groups working on this kind of personalized medicine approach. I think that, human genetics is phenomenally powerful, right? So if you look at any therapeutic, it'll sort of generally be a Gaussian, and you'll have the people that don't respond well and those that kind of, respond too well, and those that kind of fit in the middle and respond just fine. I've probably kind of diluted human genetics into a single graph, but it's important to understand that, and certainly with GLP-1 receptor, GIP receptor, there's a beautiful couple of articles in the Diabetes Journal recently where they were making the case for antagonism of the GIP receptor versus agonism when given with a GLP-1 receptor component. Yet the drugs both have the same effect, right? And they're doing two quite different things. And it comes down to possibly some human genetic support for antagonism of the GIP receptor. As in when you agonize it, that's pro-obesogenic. But on the flip side, if you agonize it for long enough, potentially it desensitizes and on top of imbalance. So I think there's tons of, really neat mechanisms at play that actually can inform other fields. But ultimately, human genetics never, sort of invented the drugs, right? So I think whilst it's useful to have, just because there's no human genetic signal for your target doesn't mean that this should be a stop-go. GLP-1 receptor agonists would have never come around if it was for human genetics, simply because there was not any major signals. There were some signals for diabetes, like TCF7L2, which was a major SNP for type 2 diabetes, and that was associated with lowered GLP-1 receptor expression in the beta cells. But otherwise, within the locus itself, not a huge amount. GIP receptor, yes, and there's tons of nice studies coming out. But I think kind of a personalized approach seems nice. But the other way of looking at that is, okay, how do we expand our drug repertoire so we can get around having too many personalizations? Because sitting in the UK with the NHS, you're going to get what you're given, right? There's no option to pay for better treatments, so it's a little bit harder to administer. But I think the field's progressing, and there's huge evidence to say, for example, some people don't respond well to GLP-1 receptor agonists, and actually you might want to put them on a sulfonylurea, which is a drug we never would want to use otherwise, because they do better. So I think it's all super interesting. I think that the human genetic angle on GPCR signaling across the board is going to be really informative for just kind of telling us kind of more about trafficking, more about recycling, all these kind of fundamental processes and how genetics influences that as well. So exciting times.
Wonderful. Absolutely exciting times coming ahead in this space. Last question. Your best advice for junior scientists who want to enter the field? That's a tricky one.
That's a tricky one, isn't it? I think just enjoy the process, right? I think there's, I've sort of got the benefit of hindsight on this, but I think you you're rushing to do everything, and kind of you've got this kind of fear of missing out. I think for sure work on other people's projects is really important, but you sort of under all this pressure to get fellowships and sort of get assistant professorships and secure your career, but just don't forget to take time. How much you're enjoying the process. I mean, again, going back to Dirk Troner, who I mentioned before, he's a pretty senior colleague, I'm very eminent, and he's told me 10 years ago he just really misses sort of that buzz of being an assistant professor when he was back at Berkeley, where he sort of had his first kind of gig, flying around presenting this kind of exciting data, et cetera, et cetera. And I think sort of as you get a little bit further on in your career, you still really enjoy it. Don't get me wrong. I really recommend it as a career. But you also just have more other stuff to do. So kind of, live for the moment sort of vibe, but also, don't be put off if you don't make it in science, because, science is a bit like a kind of a funnel, right? We put a lot in and there can only be a few come out into the faculty positions. Well, I look at my colleagues working in pharma, I look at my colleagues working in venture capitalism, I look at my colleagues working back in the clinic, and they all stay very connected to science, and they all still have a passion for science. And certainly, for me, if one of my sort of postdocs or students doesn't stay on in science, that is not at all a failure. In fact, that's just the process, the normal process. And them going into pharma or them going into kind of business, it's just really good. So don't sort of think that it's the be all and end all is what I'm trying to say, right? It's a great career, but so too are lots of other things, right? Working as a vet, do I miss it? Hell yeah, right? I mean, nothing better than wrestling farm animals on a cold day on a sort of a muddy field, right? It's weirdly gratifying. But would I want to go back and do it now? Probably not, because I don't like the cold anymore.
Thank you, David. And to your point, I think you're right. It's a funnel, especially when it comes to getting your professorship and starting your own lab. And with a PhD training and postdoc training, there's so many things that scientists learn how to do, and they can use that knowledge and those tools and skills. And I want to say maybe, head of lab adjacent positions, going into biotech, pharma, venture capitalism, scientific writing. There's all sorts of things that you can do. And no matter what, you can still, go to PubMed and read your favorite papers on your favorite topic.
Well, there's tons of people. I mean, I've always liked the technology. I would have happily run an imaging facility. I mean, it's just brilliant, right? I love microscopes and sort of everything about them. Within a university, there's loads of jobs, right? Flex manager, and these are all pretty, very respected positions, because ultimately, if those facilities stop working, then everybody has a problem. Then there's business management, there's kind of, the relationship side of things. So I just think it's a really good training. And I think in some ways, sort of having an MD or sort of having a DVM, in your head gives you a fallback, and you've got the fellowship trail, but also I bet there's a bunch of MDs who think, I don't want to go back to clinics now because I really just want to be at the lab again. And it's kind of, so I just think that there's no kind of one size fits all approach. The only thing you need to do is just make sure that you go to the lab that you want to go to, and that, the PI is the person you want to work with and has a good mentor with track record, because it's such a individual process, right? You sort of have to work with this individual every day. If you don't get on with them, then don't stay. But on the flip side, just make sure that they're going to support and look after you irrespective, right?
Absolutely. And I'll end us on a hockey analogy. And it's not that I'm not a hockey fan. I don't necessarily watch hockey. I lived in Canada for a long time, but you only miss, 100% of the shots that you don't take. So I think it's about, going out there and trying things, just as you did coming out of vet school and moving into the space you're in right now. Wonderful. Thank you, David, for your time. I really appreciate it. I had a wonderful time talking to you, and thank you again for joining me on the podcast.
Thank you so much, Yamin. It's been a great experience.
Thank you to Dr. David Hodson for taking us from veterinary school and flight sims all the way to GPCR-guided therapies for diabetes and obesity. If this conversation helped you think differently about metabolic diseases, share this episode with a colleague or trainee who needs to hear it. Make sure you're subscribed to the Dr. GPCR podcast so you don't miss parts 2 and 3 of the Celtarys research series. Don't forget to subscribe to our YouTube channel as well. And if you're listening on Apple Podcasts or Spotify, leave us a quick review. It helps more GPCR scientists and discovery teams find the podcast. For anyone who wants to unpack signaling and efficacy with real-world clarity, Terry's Corner is your next stop. Dr. Dekanakian distills the concept discovery teams rely on every day. And then check out the trailers from Terry's Corner through the link that we left for you in the show notes. Celtarys research is building the next generation of fluorescent ligands so you can track GPCRs in real time in real tissues without relying on radioactivity. We're grateful to them for supporting this 3-part series and for investing in better tools for the GPCR community. Huge thanks as well to the Dr. GPCR production team for bringing this episode to life. All the links are in the show notes, and because receptors don't work alone, neither should you. Until next time, stay safe.